Assalamualaikum everyone. So today I'm going to talk about the issue, should gene therapy be used on people? Before we go into detail, let me explain briefly to all of you about gene therapy and how does it work on human body. So what is gene therapy? According to the Medline Plus, gene therapy is an experimental technique that uses genes to treat or prevent disease. According to Your Genome Pages, gene therapy is when DNA is introduced into a patient to treat a genetic disease. The new DNA usually contains a functioning gene to correct the effects of the disease-causing mutation. This technique was first developed in 1972, but so far had limited success in treating human disease. There are two types of gene therapy. The first one is somatic gene therapy, and the second one is the germline gene therapy. So, what is somatic gene therapy? Somatic gene therapy is a transfer of a section of DNA to any cell of the body that doesn't produce eggs or sperm, and the effects will not be passed on to the offspring. While the germline gene therapy is the transfer of a section of DNA to any cells that produce eggs or sperm. The effect will be passed on to the spring. These are the techniques of gene therapy. There are three. The first one is gene augmentation therapy. Gene augmentation therapy usually used to treat disease caused by a mutation that stops genes from producing a functioning product. The second one is gene inhibition therapy. Gene inhibition therapy usually for treatment of infection disease, cancer, and inherited disease caused by inappropriate gene activity. It is to eliminate the activity of gene that encourage the growth of disease that related to cell. While the third one is killing specific cells. Killing specific cells is suitable for disease such as cancer that can be treated by destroying certain group of cells. So this is how the gene therapy works on human body. Okay, now let's look at the results of the gene therapy clinical trial on human. On September 14, 1990, W. French Anderson and his colleague at the NIH performed the first approved gene therapy procedure on a 4-year-old girl born with a severe combined immunodeficiency. It was a big success as the results of this gene therapy increased the immune function test, and she began to go to school like a normal children. And then on 1995, there are two girls that having a gene therapy with the same white blood cell having a replacement of ADA genes. Both of them increased their health and maintain the good health and stronger immune system. It shows that gene therapy can be expressed stably in white blood cell over long periods of time. They demonstrate that this Ebola virus, called the viral vector also, will not cause adverse short-term health effect. Over next 10 years, 300 clinical gene therapy trials were performed on about 3,000 individuals. However, this field was then blackened with the death of an 18-year-old male 4 days after the introduction of 38 trillion particles of recombinant adenovirus into his liver. Okay, so now we'll go to the implication of the gene therapy on humans. So the pros, it is a source of hope for a patient with the incurable disease and illness. And then the effects are long-lasting and timeless. It can eradicate disease and improve the quality of life. while the cons for the gene therapy, there is no guarantee and it can go wrong. It is costly because it using a sophisticated equipment and a high-level expertise. It is potential for developing resistance and it has the ethical concern.